Welcome back, everyone. To keep the series of incredible talks on rolling, now we begin our next session themed as Future of Diagnostics. We have Dr. Sanhita Sinhare as our chair for this enriching session. She is Assistant Professor at Department of Bioengineering, IISc. Please welcome Dr. Sanhita.
Thank you. Good morning, everyone, and welcome back. It's a great pleasure for me to introduce the keynote speaker of session 2, Dr. David Furman. Dr. Furman is the head of Stanford 1000 Immunomes Project at the Stanford School of Medicine and associate professor and director of the Buck Institute AI and Bioinformatics platform at the Buck Institute for Research on Aging. Please join me in welcoming Dr. Furman to the stage. Thank you.
Thank you so much for having me here. All I can say is, wow. There's a lot going on here in India, and this is my first time in this beautiful country. I will echo what Matt mentioned before, that there's huge potential on talent here and the vision that Longevity India is putting forward. That's my LinkedIn, that's my academic email, and I also have the Stanford affiliation. And some disclosures. The first one is Longevity India. I'm advisor on the Scientific Advisory Board. So I hate when people says aging is complex, because it implies we know nothing about it, and that's not true. First of all, aging is no more complex than the function of a cell, right? With millions of metabolic pathways happening for a particular function to be completed. And I will echo what Dr. Rattan said yesterday, that we know a lot about aging. We think it is somewhat hierarchical. So there are things that happen earlier in life, like molecular changes, cellular changes, things that have been all integrated into the so-called hallmarks of aging that you know. And then that's biological aging. There's a progression towards phenotypic aging with changes at the level of the tissues and the organs. And then the last thing we see here is systemic aging with changes at the functional level. And this is even before a disease kicks off. This is an example of phenotypic versus functional aging. This is work we've been doing with Joel Kramer at UCSF. Gray matter volume decreases with age with huge variability. That's phenotypic aging. But you also see some signs of early dementia or cognitive impairment, and that is functional aging. How do we measure biological aging? I agreed to a large extent with this previous session and what Matt said before. So you all know about DNA methylation clocks and the epigenetic clocks, and deviation from those clocks are associated and correlated with disease. That's kind of the first generation clocks that are trained on chronological age. You're trying to predict chronological age based on changes in the methylome. This is kind of a picture of how these evolved. The second generation clocks are mostly trained on diseases or mortality, so morbidity and mortality. And there's a long journey here. I'm not gonna say too much about that. I just wanna compare the different clocks, right? Here we have epigenetic clocks with high accuracy at predicting chronological age. We know nothing about interventions, how we move and change those methylation sites, CPGs, and so on. Metabolomics clocks are less accurate at predicting chronological age. We know more about the biology. We don't know how to intervene. It's very difficult to intervene with metabolic changes for those specific metabolites. Transcriptomic clocks are also noisy. We know more about biology, and the interventions are more targeted. We can do something about it. And then proteomics clocks are very accurate. Our non-biology and interventions are okay. Sometimes you select 150 proteins, so it's more difficult to intervene on such a large number of proteins. And I'm biased because we developed the inflammatory clock. And the inflammatory clock is basically a reduction to 5 proteins that we can intervene upon. And it's noisy, and I love noisy clocks because you have room for biological variation, right? If you predict perfectly the chronological age of someone, there's no biological variation there. So the immune system, which is part of what I've been doing for the past 15 years at the Stanford School of Medicine, changes dramatically with age. And we saw some of those data in the previous session. You can create clocks, like this immune clock. And many of these cells can infiltrate the tissues. This is work from Patricia Noom at Stanford, who showed that in heart disease, you have infiltration of T cells that can recognize self-antigens and viral antigens. So we had this experiment when we said, okay, if the cells from immune system are infiltrating the tissues, can we see overlap between immune genes and aging genes? So we used the GTEx, which is a large repository of public domain data sets. And we asked the question, aging genes are, say, I don't know, 1,000 or 2,000 genes. Is there an overlap with immune genes? In 51 out of the 54 tissues, there is an overlap. So this means either that the T cells or other immune cells are infiltrating the tissue, and that's what we're picking up, or the cells in the tissue are changing their identity. And that's also what's happening. We demonstrated that, and this is under review in Science. We posed the term cell autonomous immunity model of aging and disease. And so we know now that there is a deviation from the program of those cells in the tissues as we age, in addition to the infiltration of immune cells. I want to talk about inflammation. Inflammation is not the type of acute inflammation that we all know. You cut your finger, it gets red, swollen. That's acute inflammation. Let's not confuse things. When you measure IL-6, you're not looking at the right thing. It's not a zero molecule, right? Okay. So when you adjust for diseases and you adjust for things like that, typically this is not associated with aging. So we have published a paper in Nature Medicine posing this idea that chronic inflammation is very important in the diseases across the lifespan, not just in elderly adults, even in young people. And there's no canonical markers. So as part of the 1,000 Immunomes project, we decided to look at this problem more in detail. And we created the inflammatory aging clock based on proteomics data using AI, machine learning. This is deep neural network. We came up with the inflammatory age. Okay? So the previous speaker did a great job at exposing many of the immune aging features, but I think there's also a lack of extensive homework for him to see all the work that we've been doing at the 1,000 Immunomes, more than 40 publications. This is one, right? When you look at multimorbidity and the age-adjusted inflammatory age, we can predict. Frailty, this is 7 years before someone gets frail, you can predict the frailty scores based on the inflammatory age. Immune function goes down in individuals that are high with inflammatory age. So this is stimulations in many different immune cells. So this is just one plot. It took us 3 years to pipette all those 1,000 samples and do stimulation analysis in different cells of the immune system of different flavors. We know now that inflammatory age correlates with ventricular remodeling. This is in the heart, left ventricular remodeling. We know that exceptional longevity, people 100 years and older, have a lower inflammatory age on average, and we can predict mortality. So we spun out a company out of that. It was too good to keep it in the academia. We're bringing it to marketplace. It's Edifit Cell. We have this very typical business model where people can have a blood draw, and we analyze their data in our labs and give an inflammatory age index and the different proteins. And based on those proteins, there are interventions that you can actually do. And so it's not like the epigenetic clocks that there's not a lot of clarity. So that's inflammation. It's one hallmark of aging, right? Here, what I'm showing you is a huge milestone for the aging community. For the first time, WHO has recognized age-related decline in intrinsic capacity as a disease in the ICD-11. in the United States, we're still working on the ICD-10. We're way behind. But this opens the door for discussion with FDA so that we can bring diagnostics and interventions to CMS, get a reimbursement, and then these things get become free for everybody. So what have we done in the functional domain of intrinsic capacity? We have a group of 1,000 people that have been followed over the past 4 years. This is a 10-year longitudinal study of aging, where we have a ton of different functional measurements. Okay, so intrinsic capacity is based on cognitive function, mobility, vitality, hearing, vision, very basic functions. And you can see here, these decline with age, as expected. So we built an epigenetic predictor of intrinsic capacity with just one saliva sample or blood sample. You can predict intrinsic capacity in this. So pretty noisy, I love that. I'm gonna show you why I do. Almost no overlap with CPGs found in first and second generation clocks. So new, novel biology here. We show that we can use saliva samples in different cohorts to derive this intrinsic capacity predictor. We also show that it is very much correlated with immune decline. CD28 was one of the genes that are strongly associated with this metric. We have been using LLMs and with the human in the loop to get different gene sets associated with each hallmark of aging. And you can see that cell senescence and inflammation are two major determinants of intrinsic capacity. And we're predicting mortality better than first and second generation clocks. The intrinsic capacity clock is also associated with function in individual cohorts that are independent from the one that this was learned on. So say expiratory flow, hand grip strength, things like that is positively correlated. And you have negative associations with inflammatory age, walking speed, and C-reactive protein, and also smoking, and things that we know are negative for intrinsic capacity. Now we see exposome factors. So what can we do to maybe change those trajectories? We know that flavonoids are good for you. They improve intrinsic capacity. Omega-3 fatty acids of the long chain that derive from animal sources are also pushing intrinsic capacity in the right direction. Those from plants, not so much. And we have other findings there. I support very much so longevity clinics. We're working with around 18 in the US and in China. And we did this plasmapheresis exchange. So we have therapeutic plasma exchange in patients. What you see here is an increase in intrinsic capacity after the therapeutic plasma exchange was done in these patients. All right, so moving on to a more disease-specific clocks. And so this is very helpful. Intrinsic capacity is like a one big umbrella of function, but can we be more specific? We can. So accelerated biological aging in general doesn't really give you a very specific route for intervention. So what we did was to take a different approach and try and predict different ICD-10 chapters, okay? So very similar to PhenoH, you probably have familiar with this, and Brian also presented some of this in the NANES study. So we're using blood biochemistry, cell counts, aging, age and sex, do penalized Cox penalized regression, and here we're predicting ICD-10 chapters. So this means different systems, and this is a mortality predictor. So essentially, the second generation clocks on steroids, because we can now use multi-omics. We have the UK Biobank, we have the health and retirement cohort that we're bringing all into this one big study. It's under review in Nature Medicine. And I'm going to show you a lot of plots, so lots of clocks, right? The first one is in the Biobank, in the UK Biobank. Circulatory, digestive, genitourinary infection. We're predicting all those different chapters simply using standard blood biochemistry. And then the health and retirement cohort validation, you can see a very good correlation between those different predictions of mortality in the two cohorts. This is how good we're doing at predicting those mortality metrics. So this is a concordance index. We're doing quite well, for the most part over 0.8, which is quite remarkable. And if you're accelerated in the circulatory system a metric, you'll likely have angina, heart failure, heart attack, and so on. If you have an acceleration of metabolic system, you're going to develop diabetes, and so on. So these things are telling us there is biology that substantiate these claims. And if you're accelerating the circulatory system, you develop those circulatory diseases with, say, a hazard ratio of close to 2.0. But you also have high risk of respiratory disorders and metabolic and so on. So these things are also correlated. They're interconnected. They're integrated. I mentioned we're doing also the omics piece. So now we're using omics proxies for those mortality predictors. By far, the best platform is proteomics, followed by epigenetics, and then we have transcriptomics right kind of in the middle, and then the metabolomics less so. These are all the predictions, the C index of every single omics predictor of these measurements. And there's a little bit of decrease in the quality of those predictions when you use the proxies. Okay, I'm going to switch to the last 4 minutes, and then I'm going to open the stage for questions. Around 4 years ago, the Human Research Program, NASA, in Houston, reached out to me through the principal investigator there, Susie Sanello. And she told me, David, the work you're doing in the human immunomes and aging is very well aligned with what we want to achieve for our astronauts, to develop countermeasures for the accelerated aging we see in astronauts. That was mind-blowing. I didn't have no clue that astronauts suffer from accelerated aging. Most of the things that we discussed were around brain aging, cardiovascular system aging, and then muscle aging, immune decline. You have a reactivation of a variety of viruses in astronauts, so similar to what you see in humans, but accelerated, and risk of cancer. Susie, we became good friends. We got NASA grants together, started working. I get to know her family. And I understood that she really wanted to pass the torch. She was suffering from this terminal cancer, pancreatic cancer, and she passed away a year and a half ago. It's a really sad story, but I took that torch. And half of my lab started to do space medicine. And so we envisioned a way to simulate the effects of space on Earth. And this is actually available. You can buy any of these instruments. Synthicon is a company that sells this instrument called Rotating Well Bioreactor. They have the license from NASA. And so we're using this one here to accelerate the aging of different organoids, so brain organoids, immune organoids, and heart organoids, and develop diseases that are not confounded by genetics. So this is in healthy people. We, in 24 hours, simulated microgravity. We accelerate about 5 years of aging, depending on cell type. So in brain organoids, you have around 14-year acceleration. So neurons are very sensitive to this thing, and we can screen for compounds. But there's a different application, and that is for the longevity clinics, the precision longevity field, right? I take blood from any one of you. I can accelerate the aging process by about 5 years and have a window for your future aging. I can see which mechanisms will affect you and your neighbor. And so that's shown here. We took longitudinal data from the same individual 3 to 5 years apart. We took the original sample put it in microgravity, and then compared to the older sample, and did all sorts of other comparisons. What you see here is that the microgravity sample, the first one, resembles the old sample with 95% accuracy. This is the same individuals that have aged, right? So it's the real aging that happened. So how do we really bring this to the marketplace? We started Cosmica, and we're predicting and recreating, changing the trajectory of your health. And so one of the products is called Beyond Age, and we believe is going to revolutionize the diagnostics and the interventions for aging. So this is how the report looks like. It's changing a little bit now because we're incorporating one piece of data, which is very important. But this will tell you the mechanisms that will affect that person in 5-year time. We have around now 18 different hallmarks of aging that we can see today and in the future. And we have mapping with AI machine learning to different nutraceuticals that can shift that. And we tested that. So we have now a model where if you put those cells in microgravity, in the presence of these different compounds, we can see which ones will work for you, right? And so the best example of that is my friend Brian Johnson. He's taking about 100 supplements a day. He's done a great job at lowering his biological aging, but maybe only 2 are working. He doesn't know. We can actually open that window and that door. So we have many different clinics that are aligning now in this product that we're going to launch in Q3 of this year. We have partners in China as well. We're discussing at the moment with UAE, partners in Dubai, and would love to chat with you if you have different opportunities for collaborating here in India. Many of these companies are also accompanied investor. We're bringing investor money that's really intelligent money, not just any funders there. We want those that are bringing us clients. So as I mentioned, we are located at the Buck Institute. And then we have operations in China and soon in Dubai. And again, thank you so much for your time.
Thank you. Thank you, David. We have time for a few questions. Yes.
Ashwat Price Netron, fantastic talk. So I think from your microgravity studies, what you're saying is there is a trajectory you're already on, which we can amplify and then figure out what might happen. Have you seen any correlation of those individuals with their genetics or their epigenetics? The reason I'm asking is because this might be a process which is kind of difficult to do at scale, at least right now. But if there's a correlation with genetics, can you pick up signatures from there already? Where then you can correlate and say what might work or might not work.
Genetics, as in genetic variants.
Genetic variants, exactly.
We don't do any DNA genotyping.
So you've not.
We don't do it because I published a paper in 2015 in Cell that was in mono and dizygotic twins to decouple the effect of genetics and environment. We saw that 93% of the variance was due to environment. So genetics.
So then lifestyle
we don't even
is a much larger predictor of
yes
what happened to you than your genetics.
Yes. And the assumption is people, for the most part, don't change their lifestyle. That's why we are able to predict those 5 years. If you're changing dramatically your lifestyle, you have to test you again. Those trajectories will change.
And have you actually tried that out? Have you seen changes? Say, for example, I know you looked at Questine and Cohute and all these which you will recommend.
Yeah.
One, have they worked in a real life scenario?
We haven't launched yet. We don't have the data. We tested this in simulated microgravity. We added a quercetin, which is one of the compounds that was predicted to work, and you reverse 70% of the differential expressed genes in the microgravity machine. Now, what happens in vivo, we're presuming it will work, but we don't know.
Thank you.
Yeah. There's one question here.
David, it's a pretty elaborate mechanisms and then frameworks which can define a biological aging. My question is regarding microgravity. when astronaut is traveling in space, they are not only exposed to the microgravity, but other cosmic radiation. So how we are going to take account of that? That's the first question. Second question is hypothetically, if we reach to the planet which is bigger size than Earth, where gravity is going to be more than what we have in Earth, how it's going to affect the aging? Is it going to reverse it or not?
I didn't get the second question. Can you
Second
elaborate on-
question is like if we go to some other planet,
Yeah,
which is a bigger than Earth,
yeah, hugely.
will have a higher gravity there. Is it going to affect our aging process? Is it going to decrease it?
Well, yeah, great questions. I think, yeah,
Can I just add to that? Humans in our current form, are we ready to go to space yet?
right. Okay, so the question, if you guys didn't hear, is there are other factors when you do a space flight, space travel. I recommend not to do that. Don't go to space. But if you're interested in that, there is a little bit of a higher radiation. Not so much if you're within LEO, low Earth orbit. You're protected from radiation. So it's largely microgravity. It's also social isolation. It's also not moving around. That's us. They exercise a lot, right? But we showed in I didn't actually have time to show. We have 5 papers that were published all in one Nature package last year. 2 of them in Nature, 3 of them in Nature's journals, showing the resemblance between microgravity in our rotating well vessel and spaceflight. It's around 75% accuracy. So there's other factors, I agree. Stress, like how do you measure stress in a microgravity, right? But we have around 70%, 75% concordance. Are we ready to go to space and colonize other planets? I think there's a lot more research that needs to be done to put all the countermeasures ready for that to happen. I think it's a huge leap to say that we're going to be in Mars in 2030, and if we get there, we're probably going to be very, very sick.
May I ask a
Question
question? Yeah. Hi, Avi Soin, liver surgeon. I really enjoyed your talk. Now, tell us how much of the data that you have on your inflammatory clocks is lab versus animal versus human? And the human data, is it across ethnicities or is it just American?
Yeah.
See, because the epigenetic influences are very different in different populations, right?
Yeah, fully agree. This is Palo Altonians, okay? So people from Palo Alto may not be fully representative of the rest of the population out there. So for the most part, 98% Caucasians and all human, so no animal data, okay? I've tried to avoid, as much as I can, touching animals. We have to do it sometimes because reviewers request validation in animals, which is absurd, but we have to do it sometimes. FDA Modernization Act 2.0, approved in 2022, is saying no more animal work. Let's go for 3D cultures, organoid models, and so we're more in that spirit than using animals. There's another question here. Yeah. I can't hear you. Mic's not off, not on here.
Is it on?
Yep.
Actually, I wanted to know more about the telomere length. Actually, when you spoke about the astronauts, it happened to be a recent, I read a recent paper where they published that astronauts apparently have to be having the longest telomere length, but it's actually because of the accelerated aging. So how are these other companies determining, telling us that we're going to test your biological age by taking your telomere length?
Yeah.
It makes no sense.
Yeah,
Totally opposite.
I agree with you. Telomeres are overrated. That's all I can say.
It's a 40,000 rupee test and they're giving some data.
Yeah, and there's also some batch effect in those experiments that they've done in the twin study, for example. Yeah, so, and it's kind of interesting because when you return to Earth,
It's quite misleading as well.
the telomeres will shorten faster, but you have huge recovery.
Huge recovery, exactly.
Great mechanisms that are naturally put in place to rejuvenate that individual when they go back to Earth. Yes, there's a question here.
Time for one more question, maybe, and then we'll.
Yeah, Keshav?
So one thing I was wondering about inflammatory clots, have you looked at the mitochondrial dysfunction? Because being a mitochondriac, what I see by, the mitochondrial dysfunction can explain the inflammatory clot very well, because mitochondria control the innate response, the adaptive response, inflammation, and all of this thing, gas pathway. So we looked at that, and the second question was in the astronaut study. Have you looked at any difference in male and female? So we have funding from NASA, looking at how to repair their mitochondrial function, because anything we have taken from bees to mice to male or female astronauts, they all have mitochondrial dysfunction. So that's running correlation.
Yeah, great point. Mitochondria is key. I don't think it's bidirectional. The more inflammation you get, the more damage you get. The more mitochondrial dysfunction you have, the more inflammation. It's bidirectional. We've looked at many different, in different ways, the data, and it all seems to be that there's not a clear starting point. We have inflammation that happens naturally, sort of with exposome, right? And then mitochondria is also dysfunctional. The sex differences in spaceflight, there are major ones, but only from like a few examples on astronauts, because the sample size is so small. So that's why NASA called for studying more these sex differences. And we now have around 60 people that we profiled in our microgravity machine. We're going for 120 longitudinal, 10 years separated with males and females to figure out these differences. Great question, thank you.
Thank you, David.
Yeah.
For the
Thank
great.
you so much.